I'll start the recording. And yeah, so it's great to have you. If you could just introduce yourselves a little bit for our audience, that would be great.
Yes, thank you for having me. I'm Brecht de Paepe. I'm a postdoc at Ghent University at the group of Marianne de Meij at the Center for Synthetic Biology. So I'm a synthetic biologist. I mainly work with bacteria, but we are in our group also working with other types of organisms as well, microorganisms, and we're doing all kinds of synthetic biology as well as metabolic engineering.
Cool, thank you. So I invited you today after reading one of your, I guess, papers or reviews on how we need to have more standardization in synthetic biology and how we can go about that, and sort of what is the state of the standardization in the field at the moment. And yeah, I was wondering if you could sort of introduce that topic for us.
Yes, I will definitely try. It's not an easy topic, standardization. It's a very complex issue, but that will become more clear, I guess, throughout this podcast.
Yeah.
So standardization, actually, I tried to look up a nice definition, and I came across this one sentence. It's actually the striving for the opposite of trial and error. I think that kind of makes sense. That means that we aim at getting well-defined components and protocols that allow us to reuse technologies and knowledge that we gain and compare technologies and knowledge that we gain. So in all fields of science and technology, and it's actually intrinsic to, I guess, a human urge for efficiency, or you might call it laziness as well. We want to reuse the stuff that we develop. And also we want to compare the stuff that we develop, because through those two aspects, this reuse and this comparing, you can actually really minimize development times of certain technologies and minimize costs, because you don't have to redo stuff. You can build further on previously developed technologies, and you can also collaborate more easily. And this is all through this concept of standardization. And of course, in engineering disciplines, this is highly desired, because you want this efficiency, because actually your development times and costs and effort is a very important thing. You want to minimize those development times and costs. And that you can do by developing or defining components and protocols so that you can reuse previously developed components, let's say, but also compare the previously developed components that other people from other origins developed. So the standardization has a really important aspect in all engineering disciplines, and synthetic biology is one of those engineering disciplines. And also in synthetic biology, it's very important to have this standardized protocol, because within the bigger world of industrial biotechnology and bioprocess development, synthetic biology is actually the part that has the goal of making it easier to engineer life. So it's actually already in that definition of synthetic biology, let's say. It's all about engineering. You want to define certain parts, biological parts. You want to define certain biological experimental protocols that allows you to build further and faster without having to redo all this work. And actually in synthetic biology or biotechnology in general, it's even more important to have this well-defined components and protocols, because life itself is so complex that we need like a framework to work in. And that's this one aspect, the complexity of life, like additionally on the fact that it's an engineering discipline and it helps this efficiency of developing new technologies. The complexity of life on the one hand really drives this urge for standardization, as well as the fact actually that through the development of the last decades, in terms of DNA read and write technologies, we actually have this vast amount of data that we can generate. So also vast amount of opportunities that we can generate within the world of industrial biotechnology and synthetic biology. So that big pile of data that's waiting there to be processed also like really demands this standardization.
Right. Yeah. So it's really a driving force for the engineering disciplines and for synthetic biology too.
And in terms of driving force, I was thinking about this driving force aspect, and you could always bring it down to money. I think that's in all aspects of life. Well, in engineering, let's say. Don't want to overreact here, but because the standardization makes it easier and more predictable to develop new technologies and to gain knowledge about biology itself, it reduces time, it reduces costs. So that's for an engineering discipline, that's a money thing, right? So that there's an intrinsic driving force just about costs and time and effort that wants that standardization. But on top of that, I really think that the standardization also entices collaboration. So the fact that and this is still going on to this day because we're not there yet. There's a lot of work being done in the world of synthetic biology where you actually you're just redoing an experiment that someone else already performed, just to be sure, or just to be just to have it at your lab. I mean, it worked in that lab, but will it work in our lab? So that there's this because of this lack of standardization still, there's a lot of work that is being redone, actually, that can be taken away, let's say, through this standardization process. And it will allow for more collaborative work as well. And maybe a third thing, in terms of synthetic biology and standardization, I really like this concept that standardization, it implies reliability and predictability. So if you have a standardized biological system, that means that you know what it will do and you can rely on that process. So that's a great thing to hear from an industrial point of view. But I was thinking about this, and actually it's within the world of biotechnology and synthetic biology especially, the whole public opinion is also very strong, or can be strong on this topic. The whole ethical issues are very important, and we need to address these issues, and we need to communicate about these issues and questions that the public, the general public has. And I think through standardization, we actually are creating the same tools to be able to communicate better to the general public, because it will become more reliable and predictable, all these biological systems that we want to generate. So for me, that was a really maybe overlooked third driving force of the standardization process.
Yeah. And what kind of like biological components are biologists trying to standardize, for instance, or in a general sense?
If we really talk about biological components that we want to standardize, that I think that actually is only a small piece of the puzzle, but it's all the puzzles are equally important. But we talk, if we go bottom up, let's say the smallest part or biological component that we want to see standardized is our DNA sequences. And that's a classic. The logical thing to do is think about a DNA sequence has this specific function. And this relationship has to be standardized in some way. So, okay, this sequence will have this specific function. So that's the smallest component, that biological component that you can standardize. But if you go further, you use these DNA sequences to build specific devices, biological devices inside your host organism. But that you also go further, because with these devices, you can build complete circuits or complete systems and metabolic pathways or regulatory pathways inside these organisms. So it goes all the way up to the organism itself, finally, that also has to be standardized, because we use a specific E. coli strain in our lab. There's this other lab using the same E. coli strain. Then I'm just talking about E. coli, which is probably the more easy organism to ensure to have this certified standardization between labs, because it's so widely used. But if the moment you start talking about more exotic organisms, it will become a bit more difficult already. So I think in terms of the true biological components, you have these DNA sequences, devices, the organisms themselves, and actually you can add an additional layer on there, because there's a lot of work going on about microbial consortia. So you have 2 different species, you have 10 different species, all doing one specific biological process. It becomes very, very complex to have this standardized, for instance.
Could you touch on maybe some of the attempts that are going on in the field at the moment to improve standardization?
Yes. So it's a whole broad field. So you have this, because synthetic biology is an engineering discipline. That's how I see it. And this engineering discipline means that there's a design, build, test, learn cycle. It's maybe a classic way of looking at an engineering discipline, but it's an easy way, I think. And if you look at design, build, test, learn, if you look at this build aspect that you want to you actually want to standardize each of these steps in this cycle. If you talk about this building step, we talked about the parts. We talked about getting these parts into the building, using these parts and combining them into devices. So up to the organism itself. And current engineering standardization efforts are actually trying to define specific sequences. This sequence has this function, a promoter sequence, an RBS sequence, this coding sequence, terminator sequence, and even insulator sequences are being developed that try to get independence throughout these combinations of parts.
How do the insulators work exactly? Do you know?
Yes, so there are different aspects to insulators, let's say there are different ways to build them, I think. But we have some experience in-house as well, where actually you want to create a DNA sequence that is completely absent of any possible functional part. So that means that you don't want any promoter sequences in there, or RBS sequences, or transcription factor binding sites, for instance, so that if you if you have changing environmental conditions, this insulator sequence will ensure that you don't have any. If you put your insulator sequence in front of a full operon, let's say, and you have changing environmental conditions, this insulator sequence will allow you to really ensure that there's nothing else except that operon sequence being influenced by the control elements that you put inside the operon. If you don't have that insulator sequence, that means that you will have other upstream DNA that could hold a potential functionality, let's say. So these insulator sequences, they can hold some terminator sequences that has, of course, a specific function, but then you use that insulator sequence to have, to ensure that there's no read-through, for instance.
Right.
So you have your operon, and to ensure that it's not going further than it needs to go, let's say, in terms of transcription and translation.
How do you make sure that there's no sort of functional elements in those sequences? Is there a sort of central repository that you can search a sequence against, or?
This is not a standardized, of course not. Well, we're trying, but this is not there's not a standardized way to creating insulator sequences. But what you can do is you can look at databases, which are getting standardized more and more, that hold transcription factor binding sites, for instance. And then you can try to create in silico design tools that that make sure that you can create a DNA sequence that does not hold any or similar, vaguely similar transcription factor binding sites that could be in there. So,
can you perhaps like explain what are some issues with standardization of biological parts, and how easy maybe is it to standardize like small component of DNA, like for instance, like going from the bottom up?
So in terms of these biological components again, because we're still actually talking about just this build step, right? And the smallest biological component. I talked about the fact that it's logical to think about specific sequence having specific functionality. An article written in 2008 by Professor Lux, he envisions, he was thinking about even more in-depth parameters to check. So he came up with this 6 additional parameters, being independence, composability, scalability, reliability. I'm not tunability, and I think I have it all 6. I don't know, but these are all very high demanding performance requirements, and it could be that there's no true part out there that can meet these strong requirements. So if we talk about a promoter sequence, for instance, you will always have you will have to delimit or demarcate the specific DNA sequence that you think of being the promoter. This aspect itself is already variable throughout the state of the art, throughout the literature in terms of promoter engineering, for instance. On top of that, the fact that this definition, this biological definition of promoter, is actually something human, it is demonstrated by the fact that transcription and translation are not completely independent processes. So RBS sequences influence promoter sequences and vice versa. So there's a true intrinsic problem in the complexity of life, of having these defined functional parts. But actually that's not necessarily a problem, because this standardization of these parts really has to go hand in hand with characterization. So if you have a specific part defined and you create a promoter RBS coding sequence terminator, full operon, and you try to characterize, and you characterize this system in a standardized way, then you gain true knowledge about it. So you will have done a proper standardization, because they have demonstrated that, albeit difficult to really standardize the smallest biological functional part, because the fact that they're never fully independent, it is still worthwhile, and it will still allow you to do predictable building, because that's that's something that I read as well, is that standardization itself is not a goal, it's a tool. The standardization helps us to be better at synthetic biology, to reuse and to compare our work.
When you, sort of, I guess, measuring the results from these sorts of experiments where you're trying to characterize these components, is there issues in the standardization of the measurements as well?
Yes, definitely. I think that's like the other side of the coin. I don't know if that translates.
Yeah.
So this characterization itself is a very big problem, which we are actually working on in our research group as well. A colleague of mine, Lien, is working on this actually, because if you have the most used way to characterize a part, let's say, is through fluorescence, through fluorescent protein. So you will have a certain promoter and RBS. You will express a fluorescent protein, and that fluorescence is an easy way to have kind of strength, let's say, or a performance of these parts. But the whole, here comes the whole issue, I think, well, we think, is that this process is completely not standardized in none of these steps. So, and with all these steps, I mean the whole workflow. So the way it's assembled in your organism, so that's the build that we already talked about. But then you start thinking about the experimental setup, the growth medium, the device that you use to measure this fluorescence, the temperature, so all these physical parameters that you can think of, but the device itself, like something easy, like a gain or something, that all has to be, it's a big pile of metadata that has to be written down and be gathered into these reports and these characterizations. On top of that, it's very important to also think of how you process the data. So you have a certain amount of data that your device, your spectrophotometer, for instance, spews out. You then have to really think about what you want to know and how you extract these parameters and what tools mathematical tools you use. Because actually every research group is doing these experiments with, which is logic, of course, but with these with their own goals in mind. So they have this specific setup and they think, okay, I want to really know how fluorescent this strain is after 24 hours. Another person might be, or research group might be interested in how fast does this fluorescent protein degrade in this system, or how are the kinetics of this circuit. So if you have this big pile of data, then you will analyze this as well, just from your own point of view. So the fact that you can fit a growth curve, for instance, with 4 or 5 different models, and you will have different parameters being extracted out of there. Just a simple parameter as the specific maximum growth rate, for instance, will change depending on which model you use. And then we're only talking about data processing. So this is a whole process of characterization and reporting that has to be standardized as well. But there are several efforts that really, and we did some efforts as well on that within our research group, that really try to get these experiments standardized, because we know fluorescence is a very easy way to use and widely used system. We can try to start with that specific experimental setup to get it standardized.
Right. Yeah.
Right.
Are there any? Sorry, go ahead.
Oh yeah, I was just following what I've asked before about the standardization of smaller biological components, I was also wondering how far off or how easy it is to, like, for instance, standardize like a whole bacterial strain or like bigger, bigger circuits or components.
Yeah, so if we think about all the standardization efforts, so going from the biological components up to characterization, up to data management even, and repositories and databases, they actually all have to work together. They all have to be tuned to be able to have this big concert of standardized efforts going on. But in terms of that, it's also very important, I think, to realize that the whole concept of standardization is something that will evolve. So we cannot try to have a good standard for all of these aspects. We really have to aim for standards that are adaptive, so they can grow with the knowledge that we gain, because we're still learning a lot about biology, and all this knowledge will help us actually to better define these biological components or these bigger organisms. And in terms of organisms and standardizing these organisms, it's there was this big project in Europe, BioRobust, which was a very nice project, which had more than 25 partners in Europe and even outside Europe, and some small to medium enterprises as well, that really all came together to talk about standardization. And there they had this talk about the actual organisms itself that we want to see evolve into synthetic biology agents. So instead of thinking about GMOs and this, it's also very important that, like we talked about earlier about public opinion, that the right definitions and words are being used if we talk about synthetic biology and industrial biotechnology. So there's an urge to go to synthetic biology agents, and that means fully sequenced organisms, the genome sequenced organisms that are completely characterized as well in terms of growth curves and in terms of temperature optimum, pH optimum, all these physical parameters, size. How can you use it in a fluorescence activated cell sorting experiment? But there's this big list of aspects that you can think of that you will need to list and to characterize and report properly to have a specific organism to be seen as a synthetic biology agent.
If this sort of standardization were to play out as best as it could in your eyes, what would be, how would that look like if you were designing something? What would be the ideal tools, I guess?
It would be great, because the whole thing about biology and engineering biology is that DNA is actually so easy to work with. I can send anyone a piece of DNA sequence, and they can synthesize it in their lab and create their desired organism that they want. And that this is true. I think this is a true power. This digital to DNA or digital to biology aspect of synthetic biology would be really nice to really see at play at the fullest. And that's something that will be achievable when we are standardized, let's say. But it's of course a utopia, this standardization. There's no end to it, But I think that's something that we have to embrace. There's an immense value to all these efforts, to all these standardization efforts, but I also do not believe that this will ever come to an end. It also doesn't need to come to an end, or there's no end goal, let's say.
Right.
We will always be able to fine tune these standards and at all these levels.
Do you think there's anything to be learned from the sort of standardization from other engineering disciplines, or do you think synthetic biology is too different to, say, like electrical engineering or something like that?
Yeah, there's a classic comparison: synthetic biology and electric circuitry, or genetic circuits and electric circuits, for instance.
Yeah.
To learn from that, I think there are valuable lessons and there are valuable tools, I think, and maybe even legislation that we can really use, and protocols that we can really use from engineering, from other engineering disciplines that we can use for synthetic biology, but it will not be as easy, I think, because it will only hold up for certain parts in the whole range of standardization. So if we talk about data management, if we talk about data processing, those are things where we can learn more from other engineering disciplines, I think. But if we really talk about standardizing biological components, then there's no easy way. But actually, if you go indeed up to biological genetic circuits or biological devices, there are some people that already there's also already some work that really make these ID cards, for instance. You have this specific circuit, it will act like this, like an electric circuit. You can have a full ID, let's say, ID card of that specific circuit.
Okay. Yeah, great. That's all the questions I had. What about you, Sandra?
Yeah, those were all the questions that I had as well.
Is there anything else you wanted to talk about?
Yeah, I just truly think that the standardization process is something that's utmost important, but it will be very adaptive process. So it has to be something that we keep on trying on establishing. So all research groups throughout the field of synthetic biology and even beyond really have to pitch in, let's say, because then we will become a better discipline, let's say, and a more efficient discipline in the end. Yeah, well said. All right, thank you very much for taking the time to be with us today. Thank you very much for having me. Yeah, thanks a lot. Our pleasure. Cheers.